Awesome. Welcome, everyone. So I'm going to keep the introductions short, just so that Chloe and Isla get sufficient time to actually present all of their excellent work. So I'm first of all delighted to introduce Chloe for her talk. Um, Chloe is about to become a PhD alum, but she's already a U of M alum because she did her undergraduate work here and graduated from the University of Michigan with honors in 2018. She had dual degrees in biochemistry and English and literary theory. After working as an organizer for an environmental advocacy group and then as a research assistant in another lab, she joined the University of Michigan Neuroscience Graduate Program. And she joined my lab about 5 years ago. In this time, she's done some pretty spectacular work, recorded, frankly, countless thousands of neurons. She's definitely the record holder for most neurons recorded in the lab. Um, I think it's close to 4,000. Uh,
Just over 2,500.
2,500. Okay, yeah. So the total number of neurons, I think, in the database is 4,000. So Chloe's got 2,500 of those. And really has already published four manuscripts, two of which he's first or co-first author on, including one that was just accepted at Alzheimer's and Dementia. And she has others that have had revisions invited or are in preparation. As I said, she'll be defending her dissertation soon on June 5th, so I hope many of you can make it to that as well, before moving on to a postdoctoral position. And I'll let Chloe tell you about her work on retrospinal cortex and health and disease. Take it away, Chloe.
Thank you. All right, please let me know if you can't see my slides. Thank you, first of all, to the Udall Center and to Dr. Albin for this opportunity to present my work today. I'm incredibly excited to share with you results that we in the lab have recently either published or had invited to publish with revisions on two distinct retrosplenial deficits and implications for their repair in preclinical Alzheimer's disease. So just to give you some overview of what I'm going to try to cover in the next 20 minutes. First, I'd like to give you some rationale as to why we study the retrosplenial cortex in the context of Alzheimer's disease. And then I'm going to address two papers. The first looks at a cell type specific deficit in the retrosplenial cortex of the 5X FAD mouse model of Alzheimer's disease. And in addition, we look at the potential for its reversal using a classical psychedelic drug. In the second paper, which was recently accepted to Alzheimer's and dementia, we look at a cell type specific alteration in a different neuronal population, and specifically looking at an alteration in cholinergic signaling with implications for current and future muscarinic targeted therapies. And at the end, I'll take questions if there's time. So, spatial disorientation is this phenomena where someone is able to recognize landmarks that are familiar to them, but are no longer able to use those landmarks to successfully navigate familiar spaces. What you're seeing here on the left-hand side is a drawing of an individual's neighborhood after he suffered from a hemorrhage that left his left retrosplenial cortex damaged. On the right is the same neighborhood drawn by his sister. And what you can see is that he has the same or the essential environmental landmarks pointed out, but he doesn't have them oriented correctly relative to one another. I'm opening with this discussion of spatial disorientation because it is one of the first and most common deficits that you see in Alzheimer's disease. Up to 93% of people who are eventually diagnosed with Alzheimer's will have spatial disorientation appear at some point along the disease's progress. Interestingly, spatial disorientation can also result from selective injury to the retrosplenial cortex. This brain region is critical primarily for spatial navigation and spatial memory processes. So, as I mentioned 2 slides ago, injury to the retrosplenial cortex can cause spatial disorientation. And here we have case studies of two other individuals who had some sort of damage to the retrosplenial cortex and then reported profound spatial deficits. Bringing these ideas together, when we look at people with prodromal or pre-symptomatic Alzheimer's disease through PET scans, what we find is profound hypometabolism or reduced activity in their retrosplenial cortex. So here on the right is our population with Alzheimer's, here's our healthy population, and you have a reduced activity specifically in this retrosplenial area. So bringing all of these thoughts together, and because we are a retrosplenial lab, our driving question is to understand what is the cellular basis of retrosplenial dysfunction in Alzheimer's disease. We are a rodent lab, we work with mice, and so we study preclinical or mouse models of Alzheimer's disease. And just to give you some introduction to mouse neuroanatomy, this is a coronal section of the cortex. This is one hemisphere. And what you can see here, where I've zoomed in on the retrosplenial cortex, there are two anatomically distinct subregions. The dysgranular, at least at this level, looks slightly less organized. You can see a slightly denser band of cells here and then cells distributed throughout. The granular retrosplenial cortex, which is the one that we study, has this distinctive band of small pebble-like cells, hence the name granular, sitting in cortical layers 2 and 3. Now, one of our former graduate students, Dr. Ellen Brennan, took it upon herself to investigate what makes up that distinctive granular band. And what she found was that there are two main classes of excitatory or pyramidal neurons in the retrosplenial cortex. These are the more common regular spiking excitatory cell, and this is also found in other cortical regions. And then we have this low Rio base excitatory population here in purple. Now, these cells make up approximately 80% of the neurons that I showed you in that dense band, and they are radically different from an intrinsic physiology standpoint compared to this green regular spiking cell population. Everything I'm going to talk about from here on out is done in slice, so we are whole cell patchers. What I'm showing you here are traces from individual neurons that we provided different levels of current to, and you can see that they're generating action potentials in response to that current. So the key distinctions, even if you're not an electrophysiologist, that I want you to take away from this is that you can see that with small amounts of stimulus, our RS cells adapt, which means that the difference between the first two action potentials is smaller than the time difference between the last two. Whereas our LRs, the distance or the time between each action potential is approximately the same. And you can also see we call these hyperexcitable cells because at higher currents, they will fire at a much higher rate than our regular spiking population. Now, following this study, Dr. Brennan and others in the lab decided to look at the different inputs provided to these two cell types. And what they found is that there's a very distinct physical and functional separation between the types of inputs targeting these two cell populations. Our low Rio base cells get precisely targeted inputs from the anterior thalamus and dorsal subiculum, which carry egocentric spatial or vestibular input, whereas neighboring regular spiking neurons get these higher order cognitive inputs from anterior cingulate cortex and claustrum. So, given that we have a highly unique cell type that, to the best of our knowledge, has not been documented in other brain regions, and that this cell type is particularly important for processing spatial input, we hypothesized that these low Rio base or LR cells might be physiologically impaired in the 5XFAD mouse model of Alzheimer's disease. So I set about characterizing LR cells and RS cells in approximately 12-month-old 5XFAD positive mice and null littermates in the same way that I just showed you. Now, to our surprise, both of these groups of cells were radically unchanged in the 5XFAD positive mice when compared to null littermates. However, we have a tremendous postdoc in the lab, Dr. Tyler Ekins, and he recommended that we look at another facet of electrophysiology, which is the synaptic physiology. To do this, we patch the cell and hold it at minus 70 millivolts as our resting potential and wait to see these small spontaneous currents coming in. So these spontaneous excitatory postsynaptic currents, or SEPSCs, provide some insight into the connectedness, generally speaking, of a cell to its presynaptic inputs. And you can see here in the arrows, we've drawn a raw trace. You can see each of these little postsynaptic currents coming through. And when we looked at the SEPSCs of LR cells and RS cells in our 5XFAD positive cohort, we found that the LR cells had a selective deficit in synaptic input in our 5XFAD mice, whereas our RS cell population was unaffected. This was really exciting Because one of the other things that our lab is really interested in is understanding the cellular mechanisms of responses to psychedelic drugs, particularly classical or serotonergic psychedelics. And it's very well established that that psychedelic drugs can induce synaptic plasticity by interacting with the serotonin receptor HTR2A. So we hypothesize that if we have a cell type specific synaptic deficit in these LRs, it's possible that a classical psychedelic drug may allow us to repair that deficit. However, we looked at the single cell transcriptomic data, and what we found, you can see here we have a variety of clusters of the different excitatory cell types in RSG. And our LR cluster here in purple has a profound dearth of HTR2A expression. So it does not have the receptor needed to induce psychedelic synaptic plasticity. However, we have a paper that Dr. Ekins and I put out last fall in Molecular Psychiatry, and we were essentially able to show through a Cree knockout line and a few other techniques that expression of the HTR2A receptor on thalamic axons coming into RSG was both necessary and sufficient to drive an increase in synaptic boost and an increase in SEPSCs onto LRs. All of which is to say, even though the transcriptomics don't predict that LRs would be responsive to psychedelics, they can in fact experience psychedelic-induced plasticity. So with this result in mind, we gave a single systemic dose of the psychedelic 2,5-CN NBOH to our 12-plus-month-old 5X mice, and then recorded SEPSCs in LR and RS cells about 72 hours afterwards, so the drug had metabolized out of their systems. What we found is that this single systemic dose of psychedelic was sufficient to repair the synaptic deficit onto LRs without affecting the SEPSC frequency onto RS cells. Even more impressively, we took fills from the recorded cells and were able to reconstruct the neurons that I gathered electrophysiological data from. From here, one of our magnificent undergrad, soon-to-be grad students, Tao Deng, took my reconstructions and actually analyzed the morphology of each dendritic spine on these cells. And you can see them here in these orange arrows. What she found is that the mushroom and stubby types of dendritic spines, which are indicative of mature spines, both showed a significant increase in density in our psychedelic-treated LR cells. What this strongly suggests is that this psychedelic dose was able to actually induce the formation of new mature synapses onto LR cells. Now, just to recap, because I know this is a lot of information going very quickly, LR cells clearly display a synaptic but not intrinsic impairment in 5XF80 positive mice, and this can be at least partially repaired using the psychedelic drug 2,5-CN NBOH. With this very subtle effect in mind, we wanted to return to RS cells and asked whether there might be a distinct and equally complex deficit in this retrosplenial RS population. Now, in addition to psychedelics, our lab is also interested in cholinergic modulation of retrosplenial circuits. And this brings me to a really interesting cholinergic mechanism that's called acetylcholine or cholinergic persistent firing. So this phenomena, very simply, occurs when cholinergic stimulation enables a cell to fire without other input. What I mean by that is you can see a cell at baseline is getting artificial stimulation, and it fires an action potential here during the stimulus, but not after. Here, in the presence of the cholinergic agonist carbachol, the cell fires, but once this current input stops, you continue to see action potential generation. This is cholinergic persistence, and without getting too much into the details here, this phenomena is driven by the M1 muscarinic receptor. This is a metabotropic receptor. And it is also believed to be critically important for cellular working memory. Now, in a paper that we published with Isabella Yadresiak-Cape, who's our lead author on this project, last summer, we looked at persistence in LR cells and were surprised to find that LRs actually do not respond to carbachol with any type of persistent firing. And it turns out, again, when we look at the transcriptomic profile of these cells, that a pretty profound lack of the M1 muscarinic receptor and quite a significant expression level of the canonically inhibitory M2 receptor probably makes these cells indifferent to cholinergic modulation. And that's really critical per models that we developed because these cells need to be able to accurately encode angular head velocity and other spatial inputs, regardless of the cholinergic tone of the brain. However, what's really important here is that RS cells in the retrosplenial cortex do overwhelmingly display this cholinergic persistent firing. And you can see here I've highlighted the layer 5 population that we're particularly interested in, and it has a substantial amount of the M1 muscarinic receptor that drives persistent firing. And when we tested 57 cells in the RSG, 49 of these displayed some type of cholinergic persistence. So we have a cholinergic phenomena, and we know that acetylcholine deficits in cholinergic signaling are another hallmark of Alzheimer's. And we have a phenomenon that's probably critically important to working memory, which is also impaired in Alzheimer's. So we set about asking whether this cellular correlate of cholinergic working memory might also be impaired in 5XFAD mouse models of Alzheimer's disease. Now, I want to draw your attention to something that I was going to discuss in more depth shortly. But in a paper that she published back in this past fall in the Journal of Neuroscience, she looked at the transcriptomic profile of all of the glutamatergic cell types in the RSG. And in addition to our low Rio-base LR cell population here in purple, there's another distinctive population of cells shown here in layer 5A in teal. This is our layer 5A RS cell population and has also been a main focus of the latter third of my dissertation work. And we wanted to look at this cell population in particular because it does appear to be unique compared to neighboring regions. And the first thing we did to test whether or not cholinergic transmission is being impacted in the retrosplenial cortex is to determine whether or not there was a change in the cholinergic inputs to the RSG. So here we took 5XFAD positive mice and null litter mates at an advanced age and stained them for vesicular acetylcholine transporter. This is just a VACHT. It's a generally can be considered a marker of cholinergic terminals. And what we found when we analyzed the stain is that you see a significant decrease in the density of cholinergic inputs specifically coming into layer 1 of the retrosplenial cortex. Now, it's worth noting here that layer 5A RS cells also send their apical dendrites up to layer 1, overlapping with this decline in cholinergic input. Now, given this result, we wanted to look at the postsynaptic effect of the loss of cholinergic inputs. And you might predict that there might be some compensatory increase in the expression of M1 receptor, because if you can imagine you have less signal, the postsynaptic cells might want to be more receptive or more sensitive to that remaining cholinergic signal. Now, we performed the same 10x single cell RNA sequencing on our 5XFAD positive and wild type control populations. And first, we confirmed that the cell populations that we expected to be there are in fact there, and they were. And then we identified our layer 5A RSG cell cluster and looked specifically at the expression of muscarinic M1 receptor and its downstream effectors. Now, to our surprise, we didn't see a compensatory increase in expression. We actually saw a decrease in muscarinic M1 here, as well as some of its downstream effectors, including RGS4, and an increase in factors that inhibit persistence. So, as a slice electrophysiologist, what mattered to me most then was to understand what effect this loss or this reduction in M1 muscarinic receptor has on cholinergic persistence. So we took 5XFAD positive mice and littermate controls of an advanced age and recorded around 20 RS cells in each population with persistence. What we found is an incredibly profound and significant decrease in the capacity of these cells to generate any kind of persistent firing. This is an incredibly critical finding because Presently, there are cholinergic therapies that aim to mitigate both memory deficits and sleep deficits in people with Alzheimer's disease. This may suggest or may provide some beginning evidence as to why these cholinergic therapies are only moderately effective, if at all. Now, I can see that I'm running out of time, so I want to wrap up quickly. So, just in summary, in the retrosplenial cortex, which is known to be impaired in Alzheimer's disease, we have these two incredibly distinct excitatory cell populations, and each one shows a different but equally critical impairment that may or may not have a particular impact on a serotonergic and a cholinergic therapy for Alzheimer's disease. So, with that, I just want to thank again Dr. Roger Albin and the Udall Center for inviting me to give this talk. Thank you to our funding sources, and in particular, thank you to my fellow members of the Ahmed Lab. Dr. Tyler Ekins was incredibly important and instrumental in a lot of the psychedelic work. Dr. Avia was incredibly instrumental in a lot of the cholinergic work. Thank you to fellow students Elizabeth Bruins, Isla Brooks, who's now at Brown, Tao Dang, Alex Lakander, who is defending his dissertation tomorrow, and I invite you all to go see that as well. And with that, I will take questions.
Right, so I guess we have about 5 minutes for questions.
Yeah. It's a lot of information.
No, so Chloe, given the data you have, do you think there's other ways of manipulating muscarinic neurotransmission that might be more productive?
Yes, I do. And in fact, I
Hmm.
think targeting astrocytes and cholinergic recycling may be the best therapeutic path forward, particularly if we're able to intervene at an earlier time point before the M1 receptor expression is reduced. If we can basically not create conditions where you're seeing that reduction in expression, we might then be able to at least make sure that the postsynaptic cells are receptive to therapies in the short term. And in particular, I think neuroimmunology is moving in that direction.
Chloe, perhaps you said, but for the low wheelbase neurons that have the deficit in the Alzheimer mice of EPSPs coming in, um, has your lab, I'm sure you have, given the psychedelics in behaving mice and looked for spatial deficit changes in behavior?
That's an excellent question. Yes, we have. So I'll amend that just a little bit. We looked, or not we, Elizabeth Bruins, who's one of our graduate students, has looked at a cognitive flexibility task in the 5XFAD mice, and she has also looked at the effects of the psychedelics in vivo. Um, the results from that cognitive flexibility task were recently published, and I think she's still doing additional work looking at the long-term effects of the psychedelic impact of psychedelics on spatial behavior or learning behavior. Great question.
If it does work, does that mean anything about the relative importance of the cholinergic change versus the serotonin or the EPSP change?
That's an excellent question. I I think at minimum, it tells us that the system is complicated or the changes are complicated and require us to have a circuits level understanding of what is being altered. Because the fact that there were cells in the retrosplenial cortex that weren't only not changing in the 5-DOXs, but were totally insensitive to the psychedelic drug suggests that there are clearly multiple pathways that need to be repaired and might need to be repaired, addressed in their own way. Does that make sense? I would not say one is more important than the other. No, not without further evidence.
Good answer.
All right, if no other questions, thank you, Chloe, and we'll move on to Isla's talk. And Isla, if you want to go ahead and try to share your slides after Chloe unshares. So yeah, also a real pleasure to introduce Isla Brooks, who is an Ann Arbor native actually, and obtained her undergraduate degree in neuroscience from right here at the University of Michigan. Um, our lab was lucky that she took my course in her senior year, and then fortunately joined the lab as a full-time research technician for a couple of years. And where again, she was just brilliant in the lab. She mastered slice physiology in frankly weeks. Um, Rewrote and improved frankly too much code that I had written as a graduate student. She just improved all of it and rewrote it all of it in Python again in a matter of a few months and just made it all better and actually made it work all the time. And the lab still uses all of that code for many purposes. And perhaps most relevant to what she's about to talk about, she mastered a transcriptomic analysis really largely independently, completely independently, and became the bioinformatics expert in the lab. Um, Having spent her entire life in Ann Arbor, she then moved to what is actually my alma mater at Brown University, where she's currently a PhD student in Gilad Barnia's lab, studying the role of receptor expression in neuronal cell fate within the olfactory system. And despite having left the lab 2 years ago, she continues to very graciously collaborate with the lab and has now published 4 papers with us, including her first author paper in J Neuroscience last November that she won't say this, but it also got the cover, so which was great. Her first author paper ended up on the cover, so well done, Isla. So here she is to tell you all about the transcriptomic work she's done on the retrosplenial cortex.
Thank you so much, Omar. Can people hear me and see my slides? Okay, great. Chloe's talk was on a variety of angles through which we study the retrosplenial cortex, and mine will be particularly transcriptomics heavy, just to let you know in advance. But so my talk is titled "The Retrosplenial Transcriptome Across Species and Neurodegenerative Disease." So just as Chloe just mentioned, but just to go over this again, the retrosplenial cortex, or RSC, is important for navigation and memory. And it particularly helps integrate allocentric and egocentric frames of reference, that those being respectively, where is something in space and where is something relative to me. Those have to be integrated in order to navigate and remember how to get from point A to point B. Damage to the RSC causes retrograde and anterograde amnesia, as well as spatial disorientation. So you have difficulty remembering the past, that's the retrograde amnesia, as well as forming new memories, and that's the anterograde amnesia. And you have also somebody with damage to RSC would have difficulty placing the relative positions of landmarks, as I'm going to give an example of on the next slide. But another angle of interest for the retrosplenial cortex is that it degenerates relatively early on in people with Alzheimer's disease. So this is another, Chloe, when I saw yours, that was actually the one I was looking for, but this is another example of what damage to the RSC how it can present. So these 2 images from a case study where a man had a stroke that unilaterally damaged his RSC, and he and his wife were asked to place several landmarks in their neighborhood on a map. It seems like a simple task, and indeed the man's wife was able to place many landmarks on a map. Their house, can people see my cursor? I can also make it red okay. Their house is indicated with the letter A. The wife was able to place many landmarks, but the man could barely place anything on the map. And this is due just from one side of the retrosplenial cortex being damaged. So the RSC is also remarkably well conserved across species. In both humans and primates in general, as well as rodents, it's characterized by a granular and dysgranular subregion. And the RSC is always situated just adjacent to posterior corpus callosum, very medial in the brain. So in the Ahmed lab, our approaches to studying the retrosplenial cortex we employ electrophysiological, transcriptomic, and behavioral techniques to studying the function of this brain region. Transcriptomics, which is going to be the main focus of my talk, allows us to compare in a really detailed way how neurons of the RSC change across species, or how they differ across species, and how they change across disease states. And so it allows us to answer questions like how well are RSC cell types conserved between species, as well as how are different specific cell types affected by neurodegenerative disease. And relevant to that second point, our lab uses the 5-XFAD and NDUFS2 conditional knockout mouse models of Alzheimer's disease and Parkinson's disease, respectively. RSC dysfunction, as we've mentioned, is well documented in AD, and it correlates with navigational impairments. Navigational impairments are also known to present in Parkinson's, but it's not known whether the RSC is specifically related to that. So we would like to help answer the question of whether similar RSC alterations, whether to circuits or the cells themselves, underlie common symptoms of AD and PD. Given that this talk is going to be transcriptomics heavy, I figured that I would give an introduction to the workflow of single cell and single nucleus transcriptomics, as well as spatial transcriptomics on the next couple slides. So single cell RNA-seq starts with dissecting the tissue of interest and then dissociating the cells of that tissue. And then this is where the modern techniques come in that allow us to really isolate the transcriptomes of individual cells. You can barcode each individual cell such that every RNA molecule from that cell is marked with a unique identifier that allows you to tie it back to that cell, so that when you then sequence all those RNA molecules, well, technically you amplify them with reverse transcription and then PCR, you can generate a matrix of counts for every single gene in the genome for every single cell that you dissected. And essentially this matrix just looks like rows are cells and columns are genes. And that is what all of the data that I'm going to present today really originates from, just the simple cell by genes expression matrix. Also, to talk a little bit about spatial transcriptomics. So the main technique I'll be talking about today is MERFISH, which stands for multiplexed error robust fluorescent in situ hybridization. It allows you to detect thousands of cells in a brain slice and get this precise spatial information of RNA expression for each of those cells, something that single cell RNA-seq does not allow you to do. But you can only look at a few hundred genes at a time. And MERFISH specifically works, if you're curious, by assigning each RNA transcript that you want to target a unique fluorescent barcode that is read out from sequentially hybridizing hundreds of fluorescent probes. A bit of a different technique from single-cell RNA sequencing. This is a hybridization technique, whereas single-cell RNA-seq is an actual sequencing technique. Just an example of what spatial transcriptomics can make possible. These images are generated from the Allen Brain Cell Atlas, which is a massive transcriptomic atlas that includes coronal as well as sagittal MERFISH experiments for several entire mouse brains. So we can cluster the cells and color them accordingly, according to the cell type that they're a part of, as on the left image, or we can kind of zoom in and plot gene expression over these cells, as on the right, where we show, I'm showing expression of SLC17A6, which is also known as VGLUT2, an important glutamate transporter in the retrosplenial cortex. So we consulted the Allen Brain Cell Atlas in our initial transcriptomic investigations of the granular retrosplenial cortex in particular, and we found 10 main excitatory cell types in the Allen Brain Cell Atlas that make up RSG. We focused on excitatory cell types because inhibitory types like parvalbumin positive or VIP positive interneurons tend to be pretty similar across neocortical regions, and you tend to see a lot more pronounced variation in excitatory populations, especially in what defines a region. So to zoom in a little bit on these, layers 2 and 3 are dominated by LR cells, low Rio base cells, which Chloe just presented on. And these cells, importantly, are only found in RSG. Layer 2-3 IT cells, or intratelencephalic cells, are abnormally sparse in RSG compared to the rest of neocortex. You can see that just as you get into secondary visual cortex, right adjacent to RSG and RSD, there's a much thicker band of layer 2-3 IT cells that you just do not see in RSG especially. Another RSG-specific population that Chloe mentioned is found in layer 5A. We refer to them creatively as layer 5A RSG cells. And these cells express an uncommon mix of both intra and extra-telencephalic projecting marker genes. Their circuit connectivity is still not completely understood, and it's being investigated in the lab. Meanwhile, layer 6 is dominated by corticothalamic cells, which is typical, but is typical for neocortical regions, but they're particularly overrepresented in layer 6 compared to other neocortical regions within the RSG. So our next step was to look at whether RSG cell types are conserved between mice and rats. Mice and rats are both commonly used to investigate the retrosplenial cortex, with more in vitro cellular work tending to happen in mice and more complex behavioral work tending to happen in rats, although both you still see both kinds of experiments in each. We really wanted to know, do the same cell types comprise this brain region? Are we studying the same circuits in the same cells? What we found was yes. So this is just a UMAP of mouse clusters on the left and then rat clusters on the right. And what we found using a technique in which you find for each mouse cluster and for each rat cluster, we found the mutual nearest neighbors of the cells of that cluster in the other data set. And then we're able to generate a mapping of which cluster in mouse maps onto which cluster in rat and vice versa. And what we find is that every single mouse cluster has a corresponding rat cluster, with which it completely maps onto, with the one exception that layer 2-3 IT cells actually have two clusters in rats that they map onto. So there seems to be a little bit of expansion in the layer 2-3 IT diversity in rats, specifically in RSG. In both mouse and rat RSG, there are two RSG-specific clusters that dominate. So that dominate the proportion of all cells. So the layer 2-3 LR cell type and layer 5A RSG cell type make up more than 50% of excitatory RSG neurons in both mice and rats. And if you look at rats, LRs themselves alone make up 54% of all neurons in the RSG. So these cells are important for something, figuring out what. But also of interest is that both of these clusters are expanded in rats relative to mice. Another interesting fact of LRs, and this is focusing on mice again because I'm referring to the Allen Brain Cell Atlas, which is only for mice. They possess an unusual cholinergic receptor transcriptional profile. So if we plot the expression of CHRM2, the inhibitory muscarinic acetylcholine receptor, you see there's a big enrichment in RSC in general, but particularly in RSG, there's this concentration in layer 2-3, right where LR cells are. And you don't see that in layer 2-3 for the rest of cortex, not to the same degree. Whereas CHRM3, the excitatory, one of the two excitatory muscarinic acetylcholine receptors, there's actually a somewhat of a lack of expression where LR cells are. And if we just zoom in on this, you can see that there's M2 quite highly expressed in where we see where LR cells are, and M3 much more lowly expressed where LR cells are. And if we actually go in and look at the expression data from our mouse and rat data sets, this is going back to the single cell work, we indeed see that mouse and rat LR cells express very high amounts of CHRM2, and mouse specifically LR cells express low CHRM3, whereas in rat that actually has changed, even though rat still has in common the very high M2. As for how this manifests, Chloe was just talking about this, but this is data just taken. This is just a representative image from our lab website, but the paper with Chloe and Isabella is really what goes into this. That layer 2-3 LR cells don't exhibit persistent firing in response to a cholinergic agonist, whereas all of these other cell types do, such as layer 5 RS cells, which we suspect correlates to the layer 5A RSG cell type. So next I'm going to go on to talk about retrosplenial alterations in a mouse model of Alzheimer's disease. Specifically the 5XFAD mouse model. So 5XFAD mouse line is a common experimental model of AD. It specifically expresses human APP, amyloid precursor protein, and presenilin-1 with five familial mutations known to cause Alzheimer's in humans. The 3 of those mutations are in APP, and 2 of those are in presenilin-1. And in these mice, cognitive deficits tend to arise at around 3 to 6 months. It's definitely clear by 6 months, is my understanding. So we conducted single nucleus RNA-seq experiments in the RSG of 5XFAD positive, specifically heterozygous mice, because I believe the homozygous don't survive, as well as their null wild-type littermates. So as Chloe presented, the RSG cell types do remain conserved in the 5XFAD mouse line, we find all of the expected clusters and cell types that we found in our original mouse rat comparisons. And you can see each of these clusters contains cells from both the wild type and 5XFAD conditions. But when we begin to look at the specific genes that are differentially modulated between wild type and 5XFAD mice, we see signs of pretty severe retrosplenial activity deficits. So this is just a volcano plot showing differentially expressed genes in LR cells specifically in 5XFAD relative to null littermates. And if we just zoom in on a couple of these genes, NDUFA4, which is part of mitochondrial complex 4, is significantly downregulated in LR cells, which could very well be an indicator of low metabolic activity. If these cells are not firing that much in vivo, they would not need nearly as much metabolic support from mitochondria. Whereas the next 4 genes I'm going to talk about, EGR1, JUNB, FOS, and ARC, are all immediate early genes that you expect to see expressed in response to regular neuronal activity. And these are all severely downregulated in the 5XFAD mice. And again, this is just LR cells. But when we look at the general population, this is actually widespread across the RSG. So these are this is the mitochondrial complex 4 gene, NDUFA4, and then these 4 are the immediate early genes. And we see that these genes are across almost every single RSG cell type, way downregulated, severely downregulated in the 5XFAD mouse line. And just as a comparison to show that some genes actually do go up, this is GRM1, which I believe is metabotropic glutamate receptor 1, potentially showing a compensatory mechanism to increase sensitivity to incoming glutamatergic neurotransmission if they're not getting much activity in the first place. Focusing now on layer 5A RSG cells, Chloe went over this as well, but we see signs of altered cholinergic persistent signaling in these cells. So just to go over the components of the cholinergic persistent signaling pathway, it starts with signaling at the M1 or CHRM1 receptor, which binds to GQ. A class of genes called RGS genes, or regulators of G protein signaling, actually inhibit GQ, or they help modulate it so that you don't get too much activity. And then TRPM4 is the downstream ion channel thought to result in persistent firing. When we look at layer 5A RSG cells, we see downregulation of CHRM1, as Chloe mentioned, and upregulation of TRPM4, although TRPM4 is expressed at such low levels that it's difficult to get a significance measure of this upregulation. An interesting fact to mention, though, is that human CHRM1 has previously been shown to be downregulated in both AD and in both Alzheimer's and Parkinson's. When we look at the overall trend of RGS genes, we see downregulation with some exceptions, but specifically RGS4, the most severely downregulated RGS gene, is known to interact with G-alpha-Q specifically. So referring back to our persistent signaling diagram, there's a reduction in M1, a reduction in RGS potentially to compensate, and an increase in TRPM4 also potentially to compensate for the reduction in M1. But reduction in M1 and decrease in RGS and increase in TRPM4 predicts a mixed overall effect on the persistence pathway. And as Chloe presented, this actually manifests as a decrease in the percentage of layer 5A RSG cells that are able to persistently fire in response to a cholinergic agonist. Something to keep in mind with cholinergic therapies for Alzheimer's is that they may not work well in patients with this receptor deficit. If there's no receptor to target, these therapies may not be effective. So I know I'm beginning to run out of time, but I'm just going to present some initial findings from our Parkinson's mouse model. Well, not our, but the Parkinson's mouse model that we've been beginning to study. So this line is a conditional knockout of the mitochondrial complex gene NDUFS2. So LOX P sites flank NDUFS2, and then Cre recombinase is driven in this line by dopamine transporters. So specifically, dopaminergic neurons experience a metabolic deficit because their mitochondria are missing a specific part of the electron transport chain. So this line, these mice model Parkinson's disease in several key ways. So they have progressively deteriorating motor capabilities. They actually show therapeutic response to levodopa at early but not late disease stages, similar to human Parkinson's. And they exhibit pausing in their gait, which models freezing of, that's meant to say GAIT, in humans. Although one caveat is that these mice do not survive beyond 5 to 6 months. We conducted single nucleus RNA-seq experiments in roughly P80, postnatal day 80, NDUFS2 conditional knockout mice, and as well as their unaffected siblings. And this is just showing from the paper that first documented this mouse line that at P30, there is a therapeutic response to levodopa in a Y maze task where it successfully rescues behavior, but not at P60. So it's pretty significant change just between P30 and P60. Some initial work from studying this mouse line going on in our lab is that we see specific gait deficits in NDUFS2 conditional knockout mice, where let's see, there are shorter individual step distances in the knockout, as well as longer time spent on while walking, longer time spent on diagonally opposed paws. This is also true for the fore and rear paw pairs, but there's a clearer effect for the diagonal paw pairs. And this may reflect difficulty coordinating gait and needing to balance more. So when we look at specific cell types that are affected in the transcriptomics in this model, what we see is that LR, and I'll first focus on the corticothalamic cells, show deficits in these immediate early genes, the same ones that I was focusing on for the Alzheimer's line. So these are our 2 layer 6 CT clusters. They both show decreases in these 4 immediate early genes. And then this is just the same thing for LR cells. But then when we look at the layer 5 RSG cells, we don't see much modulation at all. So this is a cell type specific deficit in activity that we believe we're observing based on the expression of these immediate early genes. And it specifically seems like the RSG thalamus circuits are what are being impaired. So Chloe presented this figure as well, but the LR cells, just to remind everybody, the LR cells are connected, receive thalamic inputs, and the corticothalamic cells that are being affected are the ones that project to thalamus. And those are the cell types that we see having what we believe are activity impairments, but not, interestingly, the layer 5A are RSG cells that do not receive thalamic inputs. And this is just showing the general population of, or this is showing these 4 genes across all clusters. But again, layer 5A RSG is not really affected, whereas LRs and the CT cells, as well as layer 6B, although I didn't go as much into them, are. What we can do now that we have transcriptomic data sets for both the Alzheimer's and Parkinson's lines is we can begin to look at shared as well as different transcriptomic changes in each of these models. So this is just a kind of a combined volcano plot showing the log fold change on the y-axis. More positive means a greater increase in the NDUFS2 Parkinson's model. And on the x-axis, more positive means a greater increase in the 5-XFAD Alzheimer's model. So just to orient people to the quadrants, upper right means a gene is going up in both the Alzheimer's and Parkinson's model. The bottom left is going down in both of these models. And then these two quadrants are up in one, but not the other. Just to give examples of a few specific genes. So mammal2 is important for notch signaling, and that we see going up in both. IQ-gap2, which is going down in 5XFAD, but up in the Parkinson's model, is important for cell-cell adhesion. EGR1, there's an immediate early gene that we focused on before, is shown to be down in both of them. And then NTNG1, here on the bottom right, is down in the Parkinson's model, but up in 5XFAD, and that's important for axon guidance. So we're still beginning to parse out. This is very new. These are very new analyses that I haven't had a chance to really dive into yet, but we can now compare and contrast the gene differences, the expression differences that we see between these two models. And just to give a broad overall statistic, within LR cells, 70% of significantly differentially expressed genes change in the same direction in the 5XFAD and NDUFS2 lines. For layer 6 CT cells, which again we showed to be modulated in both, that's 73%. But for the layer 5A RSG population, it's only about 50%. So we're seeing some cell types affected similarly by these two models, but others quite differently. And that's something that we really need to dig into. So just to summarize, we've established a cross-species transcriptomic atlas of RSG cell types that we can now use when looking at disease conditions. We've confirmed that mice and rats have the same underlying cells. We've shown that 5XFAD mice have cell type specific disruptions of cholinergic signaling, with fewer layer 5A RSG neurons able to persistently fire in response to a cholinergic agonist. We've also seen that the RSG of both the 5XFAD and NDUFS2 mice exhibit signs of severe neural activity deficits, and with the expression of those immediate early genes. The 5XFAD deficit, however, is more general and widespread, whereas the NDUFS2 appears to particularly affect the thalamus-connected cell types, whether they're pre or postsynaptic. And we're just beginning to look into how these AD and PD models share transcriptomic modulatory effects depending on the cell type. With that, I would just like to thank the Ahmed Lab and everybody who has worked on all of these projects. So thank you to Omar and the lab for continuing to include me in collaborations and for inviting me to give this talk. And thank you to Isabella, especially for meticulously performing all of the dissections that make all of the transcriptomic work possible. None of this would be possible without her careful dissections. And then thank you to also Chloe and Tyler for all the EFIS contributions to everything and helping figure out the puzzle of the RSG. With that, I'm happy to take questions.
Brilliant. Thanks, Isla. Questions?
Yeah, so quick question. So that was really nice. So how much of this do you think could be driven by a change in afferent activity from the thalamus?
Specifically, the reduction in immediate early genes that we see in both
Yeah,
models.
that's what that would seem to be like, I would think, right?
For the cell types that are particularly driven by thalamus, like LR cells, which are directly postsynaptic to anterodorsal and anteroventral thalamus, I would absolutely believe that would be the main driver. But for other cell types, I think it's a little bit more complicated. Like the layer 5A RSG cell type, we don't see any evidence of an activity deficit, at least through immediate early genes, in the Parkinson's model. So while, yes, I believe that could drive the changes that we're seeing, and additionally, that doesn't explain why we see a broad downregulation of these immediate early genes in basically every single RSG cell type in the 5XFAD model, even though some of those are not directly driven by thalamus. So I think that it definitely has a part to play, but I'm not sure it's responsible for everything.
Just to follow that question up, Roger's question up, Isla, do you
Yeah.
think then, consistent with other models, other results emerging in other Parkinsonian models, for example, the MyroPark model, that thalamic deficit may play a bigger role in Parkinson's models than in Alzheimer's models, just to kind of put together what you and Roger were just saying.
Yes, that would absolutely make sense. That perhaps thalamocortical dysfunction may be how
Hmm.
disruption of dopaminergic signaling makes it to the cortex. Yes.
Great.
I actually have a question if no one else is jumping in.
Yeah.
I'm very surprised that you're not seeing changes in the layer 5A RSG population, particularly because those cells should be getting inputs from both LRs and CT cells in the same region.
I see.
So if these are both, I mean, deficient, if those cells are both impaired, why are you not seeing an impact in the layer 5A population, do you think? And just as a follow-up, because I know that we talk a lot about whether or not the cell populations that we get are representative, do you think that there are layer 5A cells that have died off that show vulnerabilities that the remaining layer 5 cells that you have analyzed are not showing? And if so, how do you then interpret that? Sorry, that was a three-part question, but
Okay. Just to make sure I understand. You're what you're saying, why don't we see changes in the layer 5A RSG cells because they're postsynaptic to these other cell types that are being downregulated? Um, I'm not sure. I I've yet to look into receptor differences in the Parkinson's line that could explain perhaps they're actually successfully compensating for a lack of or lower synaptic input by overexpressing glutamatergic receptors. But I will say that the overall, even though we do see cell types that have downregulation of immediate early genes in both models, the deficits are far more severe in 5XFAD. So that may be the 5XFAD model may simply be too much to compensate for across the circuit, whereas this model is much more targeted. It's knocking out dopamine, or not knocking out, it's impairing dopaminergic neurons' metabolism. And that's a very specific effect, whereas as opposed to expressing, problematic proteins in every neuron in the brain. So I suspect that just due to the specific targeting of the specific modification that is being made in the NDUFS2 Parkinson's model, it's some circuits are able to compensate, whereas in 5XFAD it's too general. Yeah.
thanks.
And I'm sorry, were there other parts of your question? I focused a lot on the first part.
I think we're out of time, so I can ask you
Okay.
afterwards. It was more about like representative populations, but
Okay.
yeah.
Okay, well, I think we're at the end of the hour. I want to thank Chloe and Isla for two really excellent and very interesting talks with just a really nice spectrum of work. So thank you. And thanks to Omar for his emceeing as well. So,
Anytime. Thank you, Roger.
all right.